All right, so this is going to be a summary of what we've covered in class on making ATP. So first, let's talk about the difference between substrate level phosphorylation and oxidative phosphorylation, because we're going to be looking at the different processes, and notice we're going to be looking at how many ATP are made by each of these ways of making ATP. So first, substrate level phosphorylation. So this is substrate level phosphorylation. This is a substrate, this is an enzyme, and this is ADP. And what literally happens is an enzyme comes in and it connects the ADP to the phosphate. So it phosphorylates ADP by just transferring from a substrate a phosphate. The other kind of phosphorylation is called oxidative phosphorylation, or it's also called chemiosmotic phosphorylation. Both of these names match what it is. First of all, oxidative does not mean oxygen, although we are going to learn that oxygen is involved in us in this type of respiration. Oxidative refers to the fact that it involves redox reactions, which is basically electron transfers from one thing to another. And chemiosmotic refers to the fact that this is going to be across a membrane, but this is a membrane, and it's literally going to involve transfer of, in this case, it's going to be hydrogen ions. We're talking about a difference in charge and a difference in concentration. We're going to have hydrogens being built up on one side. We're going to have electrons moving down through these proteins. And this is going to involve electronegativity. It's going to involve redox reactions. Oxygen is going to be waiting over here on the other side. And it's going to sort of be the pull to pull all of this towards it. And that's going to make water at the end. This can also be done with sulfur. Certain bacteria do this with sulfur. So it's not the fact that oxygen's involved, it's the fact that redox reactions are involved. And the ATP is actually going to be made not by the actual transport of electrons, but by the flow of hydrogens, which are actually at this point protons, because they have a positive charge and hydrogen has only one proton and one electron. So if the electron's gone, it's a proton. But this is the enzyme called ATP synthase, and this is going to actually generate the ATP. So, or connect, I should say, connect ADP to phosphate. But we're going to come back to that in detail in a few minutes. So let's go back to this chart. All right, so it turns out that glycolysis makes a net of 2 ATP, and it makes them by substrate level phosphorylation. Oxidation of pyruvate doesn't make any ATP. And the Krebs cycle is going to make an additional 2 ATP. So our total ATP made by substrate level phosphorylation is going to be 4. Oxidative phosphorylation we're going to talk more about at the end, but oxidative only occurs in the membrane of the mitochondria, and in a perfect world we're going to say that it would make probably 32 to 34 ATP. We're going to go with 32 here. And when you add these two together, this is what's going to give us our total of 36 ATP that we learned at the very, very beginning of our lecture. Now, let's go to another slide, and let's do a little walkthrough showing where these ATP are being generated. So, we started with glucose, and glucose has 6 carbons. Our first step in the cytoplasm was glycolysis, the splitting of glucose. At the end of this process, glucose has basically been broken in half into 2 pyruvates, and pyruvates are 3 carbon molecules. So here's our 2 pyruvates. Now, it actually took 2 ATP in this little energy investment phase, so ATP go in, but we make 4 ATP in the second part, the harvest phase. And this is why we come up with a net of 2 ATP being generated by substrate level phosphorylation when glucose is split into pyruvates. Now, those pyruvates, what's going to happen next is those 3 carbon pyruvates are going to lose a carbon, becoming what's called an acetyl group, which is 2 carbons, and that acetyl group hooks up with a coenzyme that we call coenzyme A. So we make 2 of these because we had 2 pyruvates. So each pyruvate is becoming acetyl-CoA, and these are 2 carbon compounds. No ATP was made here. And now each of those is going to enter the Krebs cycle. Usually they draw the Krebs cycle once. I'm going to draw it twice just to show you that now this 1 acetyl-CoA, and it's 2 carbons, not 2 acetyl-CoAs, is going to feed into the Krebs cycle, and when it does, what you're going to see is it's going to generate, by substrate level phosphorylation, 1 ATP, as is this acetyl-CoA. It's going to also. So our total from the Krebs cycle is 2 ATP. So this is where our 4 ATP from substrate level phosphorylation are coming from. 2 of them here and a net of 2 of them there. The rest are all going to be made in the end in the electron transport chain. I'll just put ETC, which is somewhat complicated, but bottom line is I'm going to show you how we can generate approximately 32 here. So that's our overview. Now let's go back to the chart and let's take a look at carbon dioxide. Now glycolysis doesn't make any. And the easy way to remember that is all that happened was glucose, which was 6 carbons, got split in half. We didn't lose any carbons here, so there's no way carbon dioxide could be generated. Oxidation of pyruvate, though, generates 2 carbon dioxides. The Krebs cycle generates 4 more carbon dioxides. And all the carbons from glucose are gone at this point. If you add that up, that's 6 carbons. Glucose was C6. So all of our carbons from glucose are going to be gone at this point. 6 CO2, which is all the carbon dioxide that's made. So now let's go back to our breakdown here and look at where the CO2 come from. So here's our 3 carbon pyruvate, and it broke into an acetyl-CoA, and it lost right here a CO2. And the same thing's going to happen to this pyruvate. It's going to also lose a CO2. And now the acetyl group only has 2 carbons. And literally all that's going to happen to our acetyl group through several reactions is our 2 carbon acetyl, each of those carbons is going to split off, and we're going to get from the 1 acetyl group, which was 2 carbons, to 1 carbon dioxides. So 2 CO2, 2 CO2, and that's where we get our total of 4 carbon dioxides. Notice, and this doesn't add up quite properly because this is a redox reaction and water's involved on both sides, and our overall reaction is a net reaction. But the bottom line is, what I want you to take away from this, don't get too bogged down in adding up why the oxygens don't quite add up properly, but in theory, all of our carbons and all of our oxygens have been converted to CO2. I know there's only 6 here, but again, it has to do with the redox reactions. The oxygen you breathe has not come into play yet. This O2 is still sitting there. Which brings us to, where did the hydrogens go? We had 12 hydrogens here from glucose. We know that at this point, the carbon and oxygen are gone. That's waste, you breathe it out. What happened to the hydrogens? So let's go back to our chart one more time. The hydrogens are being picked up by these coenzymes. NAD, you're not making NADH. What you're really doing is hooking a hydrogen to it. And the same thing is true with FAD. That was a little sloppy. It also is picking up hydrogen. Again, the numbers aren't going to add up perfectly if you say FAD and now it picks up 2 hydrogens, but again, don't worry about that. All right, so where do these pick up the hydrogens? In glycolysis, we made 2 of these. Oxidation of pyruvate made 2 more, and the Krebs cycle makes 6. They're going to actually go back to being NADs again. They're going to get rid of their hydrogens when they get to the electron transport chain. So we've made 10 NADHs at the end here. That's the reduced form. And then the FAD's only in the matrix of the mitochondria. So in the Krebs cycle, we're also going to generate 2 of those. And so that's a total of 2. Notice water has not been made yet in our net equation. And oxygen that you breathe has not been used yet in our net equation. All right, so coming back to here, let's talk about the NADHs. I'll use a different color. All right, so we made 2 NADH here. Then we made an NADH for this pyruvate and an NADH for this pyruvate, so that's why we say we make 2 here. We said we made 6 here. We actually make 3 from one acetyl-CoA and 3 from the other acetyl-CoA. And then we also make 1 FADH2 and 1 FADH2. So this is where our totals are coming from: 6 NADHs and 2 FADH2s. All right, so at this point, all of our hydrogens are trapped in the coenzymes. All of our glucose, our carbons and oxygens from the glucose have been converted to carbon dioxide, and all that's left is the electron transport chain. So if I go back real quick, this is in the net equation. We make our 6 waters here in the electron transport chain, and this is where our oxygen is actually going to come into play. So let's go over to a new slide and look at just the electron transport chain. So if this is a mitochondria, glycolysis happened out here. It made 2 NADHs. Now, it turns out that in most cells, although there are a few, liver cells and some others, these don't actually cross the membrane. They are made in glycolysis, they're recycled in the cytoplasm. So what they end up doing is they actually transfer their electrons here to FADH. It's going to be important when we add up our totals. So technically, we're going to pretend like these can't pass. They're going to give their electrons to FADH. All right, in the matrix is where we had the Krebs cycle. The oxidation of pyruvate happened here, and now we're at the electron transport chain, which is going to happen here. So let's take a look at the electron transport chain really quickly. So let's pretend like this is the inner membrane. Let's say that this out here is our outer membrane. And now here comes all of our NADHs and our FADH2s. And NADH and FADH2 at this point are going to give all their hydrogens to this series of proteins in the membranes. This is an awful drawing of it, but it's not really important for our class. So imagine that our hydrogens are all getting dumped off out here in this intermembrane space. But notice these are hydrogen ions. Their electron is missing. That's because the actual electron is traveling down what we call the electron transport chain. All the way at the end here is going to be our enzyme. This is ATP synthase. This is what actually makes the ATP. And what causes the ATP to be generated is, imagine that oxygen's waiting over here. She's very attractive. She's very electronegative, and she's pulling the electrons and the hydrogens towards her. So we have an electrochemical gradient, which we learned about in a previous chapter. Here comes our hydrogens, and they're going to travel through ATP synthase. The electrons and the hydrogens are going to hook up with oxygen, and we're going to end up with our water. All right, there are some cells. I started to draw this here. There are some cells in organisms that actually have a different protein here. If the hydrogens don't go through ATP synthase, they're still going to release their energy, but it won't be captured by ATP. And so this is going to actually generate heat. And so those cells are found in Arctic organisms and some babies, and it generates heat for them. You generate heat regardless. That's why you shiver when you're cold. Remember, a lot of the energy is not extracted. All right, for every NADH that enters the electron transport chain, we're going to actually generate 3 ATP. And for every FADH2 that enters the electron transport chain, we're going to generate 2 ATP. Not directly, but through the path. All right, so first we have glycolysis. Glycolysis made 2 NADHs, but with the exception of some particular cells in the liver, these can't pass into the mitochondria. They actually pass their electrons to FAD, and so they only generate 4 ATP in the electron transport chain. 2. Next, we made 2 NADH in the oxidation of pyruvate. That's going to make 6 ATP. We made 6 NADHs in Krebs. That's 18 ATP. We made 2 FADH2s in Krebs. That's going to generate another 4. If you add all that up, you're going to get 32 ATP. Add in the 4 that were already made, and that's our 36. So that's it. Sometimes you can get 38 because, like I said, there are some cells that can allow this to transfer to the mitochondria. Also, in a perfect world, you'd make